Hi everyone, it's noon. So we're going to get started on our one of our, I think our third or fourth GrowON webinar for 2022. And Siobhan's going to talk a little bit more about our webinar series.
Great, good afternoon everyone. Thanks for joining us. Our GrowON webinar series we launched in 2020 and we've been working on it for a while. So it includes topics on both ornamentals and vegetables, as well as vertical farms and cannabis. It's led by our FRAC greenhouse team, and you can see all four of us on the screen there. So Sarah and I on the ornamental side, and then our colleagues Kara and Patty on the vegetable side. So we talk about things that are related to production, things that are related to IPM, and you can usually always find a topic that might be of interest to you. We try and host these webinars on a monthly basis, so you can get the latest news on what's upcoming in our webinar series on our blog at onfloriculture.com. So before we get into today's webinar, we're talking about botrytis today. It's a really important topic, and we have a poll before we get started. We want to know, how does botrytis impact you on your farm? So please let us know which types of crops you have issues with botrytis in. Is it cut flowers? Is it more of a potted crop? Is it in your vegetable crop or cannabis?
Starting to see some answers come in now. Vegetables and herbs was in the lead, but as people are answering, it's shifting. We've got some cut flower answers and some annual answers as well. We'll give that a few more seconds as a couple more people are joining. I was betting on cut flowers, so so I'm waiting for a few more cut flower people to join. All right, okay, well, we'll end that poll. It's a good mix. Cut flowers, annuals, vegetables and herbs, and other seem to be our biggest categories.
Great, thanks for taking the poll. That helps us make sure that we're targeting the right people when we start to make resources for you. So today's webinar is part of an ongoing series that we have going with our friends over at Michigan State University. So joining us today is Jeremy Jubenville, who's an extension coordinator with Michigan State, and I'll toss it over to him to take over.
Yep, that's right. Thank you, Siobhan. Hello, everyone. I first want to thank Sarah and Siobhan for graciously allowing us to share the airwaves this afternoon. Very, very gracious. And so as Siobhan mentioned, my name is Jeremy, and I am a greenhouse agent with Michigan State University Extension. In my role, I work primarily with commercial flower growers across 7 counties in Southwest Michigan, based in sunny Kalamazoo. Before we start, I want to bring your attention to the Q&A button. It is located at the bottom of the Zoom window, or it should be. Please feel free to submit questions at any point during the presentation. We will be keeping track of them, and we will be sure to give our guest a chance to answer them at the end of the presentation. So here at MSU, we have a great group of people on the ornamental horticulture team, and I am pleased as punch to be able to introduce our special guest this morning. So Nikki Lukasko is a PhD student in Mary Hausbeck's plant pathology lab. I first met her 4 years ago when she was brand new and collecting samples of botrytis from greenhouses all over the southern half of Michigan. And she tested these samples for fungicide resistance. And as you might expect, one has to be meticulous when working with microscopic organisms, and this was painstaking and time-consuming process. We are very appreciative of all of her hard work, and we're very happy that she can share the results with us here today. Nikki, thanks for joining us.
Thank you for the nice introduction, Jeremy. I'll see if I can share my slides correctly now. Does that look all right?
Beauty.
Great. So as Jeremy said, my name is Nikki Lukasko, and I'm a PhD student in Dr. Mary Hausbeck's lab at Michigan State University. You see Blair Harlan's name is also listed here. He's a research technician in our lab who has run a lot of the fungicide efficacy trials that I'll talk about in a little bit. Today, we're going to be talking about battling botrytis in the greenhouse, environment, nutrients, and fungicides for disease control. So to give a little bit of an overview, first, we're going to talk about classic botrytis symptoms in ornamental greenhouses, some of the target crops that are most susceptible to botrytis. Then we'll talk about how you can alter the environment in the greenhouse to lower disease pressure. We'll talk about conventional fungicides and biopesticides, and this will include both best practices and product recommendations towards the end. And then throughout this, I'll talk a little bit about the ongoing research that I've been doing looking at fungicide resistance frequencies and the potential for calcium as a disease management tool for botrytis. So moving on, here is a picture of the characteristic fluffy brownish gray masses that have given botrytis the nickname of gray mold. Gray mold, aka botrytis blight, is caused by the fungal pathogen Botrytis cinerea. This is a great example of it on New Guinean patients, which is one of the most susceptible floriculture crops to botrytis. So moving on here, now we're looking at another susceptible crop, zonal geraniums. Like I said, these are what we call magnet crops, or those that tend to get botrytis first and sometimes have the worst symptoms. In the case of geraniums, once these plants start to flower, the petals drop on leaves that are lower in the canopy. You can see this picture on the left and right a little bit closer. These flower petals here provide some of the sugar that's easily available for the fungus and really gets the disease going. And at the same time, this picture on the left here shows you that botrytis tends to take place lower in the canopy, sometimes sitting on top of the soil surface. This humid microenvironment is really ideal for infection. So next here, we're looking at petunia, which of course is another extremely susceptible crop. Like petunia and a lot of other bedding plants, and unlike geraniums and some of the other crops we'll talk about today, infection can take place at earlier stages and really any stage in petunia's life cycle. Unlike poinsettia or geranium, you can usually just remove infected portion of the plant or toss that plant. But in the case of bedding crops, sometimes you can lose a whole flat or at least the whole plant. In the same case as geraniums here, where flower petals tend to provide that sugar that really gets infection going. You can see petunia meltdown here on the right side, where that flower starts to decay, becomes really susceptible to botrytis, and can serve as an inoculum source. So the last of the crops I'll be talking about today is poinsettia. In the case of poinsettia, bracts tend to be very susceptible to botrytis infection. And in addition, these plants are grown in the fall when you have weather that's especially conducive. Botrytis as well on days with high relative humidity and overcast. So now looking at a little bit of the biology of botrytis. Like I said before, botrytis is also known as gray mold due to the large number of gray spores it produces. So if you look at these pictures on the bottom left and right, here we're looking at gray masses of botrytis. And if you zoom into these, what you see is what the picture in the middle. That's a picture of Botrytis cinerea under the microscope. Kind of looks like a tree growing leaves or maybe a cluster of grapes. These spores are airborne, spread throughout the greenhouse and air currents. Sometimes if you have a cluster of spores on the left or right here and that plant pot may get bumped, these spores act sort of like dust and rise into the air and can move around the greenhouse easily. When that spore lands on a leaf and the environment is humid with free moisture, and ideal for botrytis, it infects, produces more of this on the surface, and this is the process that we see in the greenhouse over and over. So what do we need for this disease to take place? So for botrytis blight, you pretty much always have Botrytis cinerea present in your greenhouse. The fungus is ubiquitous, but you're only going to see disease when you have ideal environmental conditions. So this includes the high relative humidity that I will say over and over. Botrytis really likes humidity above 85%. So if you can keep it below that, you'll lower disease pressure. It needs free moisture in the greenhouse to cause disease. Botrytis tends to prefer older tissue and dead plant material. Unlike a lot of other plant pathogens like your mildews, for example, botrytis doesn't care if your plant is alive or dead. Dead plant sitting in the corner can be a great inoculum source for botrytis. And as I said, spores are spread through air currents. So here now we're going a little bit into the cultural disease management practices. Number one here is going to be to scout as often as you can, especially under the canopy of what I said before as magnet plants. So this is your impatiens, your geranium, your petunia, your poinsettia. As mentioned, this fungus is known for its ability to produce millions of spores very quickly. Just a couple of leaves can have several million spores on them. It means that outbreaks can occur quickly as a result of just a few infected leaves. And this disease often starts underneath the canopy, as you see in these photos. So like under the canopy of geranium, under the canopy of poinsettia, often right on top of the soil surface. If you don't have much time to scout, start by looking in the most humid and damp area within one of your most susceptible crops. And if this area is clear of botrytis, it's likely that others are clean as well. And it's important also to keep in mind that symptoms can occur just 24 hours after infection takes place. So scout as often as you can. So moving on now, we're looking at spacing your plants out to lower relative humidity. I can walk into a greenhouse and pretty much tell based off how it feels and looks right away if there's going to be a lot of botrytis in there. You see this photo on the left, a lot of plants really cramped together. I'm sure that under all of these leaves, right on the soil surface, you're going to find botrytis throughout. This is going to create that microenvironment right under the canopy that's ideal for botrytis. So if you space out your pots like you see in this picture on the right, especially as your plants get older, it's going to help keep disease pressure down. So next here, we're looking at keeping your moisture levels as low as possible. And this may mean preventing standing water in the greenhouse, as you see here on the left. That's only going to increase your humidity as well. Watering earlier in the morning means that you're allowing for extra water to evaporate. Again, that needs water to germinate and cause infection. Venting or heating your greenhouse and installing fans, really keeping the air moving, will allow plant material to dry out faster and leave it less prone to botrytis. And as I said, remove those diseased plants as soon as possible. If you see this photo on the bottom right, you have a bunch of healthy pots of petunia right next to a few that are completely dead. I guarantee that these pots over here on the right have botrytis, and the longer this sits here, the more likely botrytis is going to spread throughout your greenhouse. So remove those diseased plants and trash as well from the production area, as both of these can serve as inoculum sources and be a cause of future outbreaks. So why do we do all of this? Keeping your greenhouse as dry as possible, lowering the humidity and all of that is important because these spores can germinate in just 3 hours. You can have infection take place in 6 to 9 hours, and visual symptoms just 24 hours after that. So disease occurs very quickly. Environmental manipulation is really one of the strongest tools when it comes to disease control. So knowing all of this, even after all these cultural management practices, fungicides are often necessary for botrytis control. Applying these fungicides at effective rates is super important. The lowest rate on the label is the lowest rate that can still provide effective control. If you see that the lowest rate is 4 fluid ounces and you go for 2 fluid ounces instead, it's possible that you won't get any level of control. So stick to the label always. And this includes following the application intervals on the label. Some of the slides I'll go through later today, we're going to compare 7 and 14-day intervals. We'll talk about the importance of alternating effective fungicides for resistance management next. So to give a little bit of background, if you're unfamiliar with FRAC, FRAC stands for Fungicide Resistance Action Committee, and this is a group that works to prolong the effectiveness of fungicides liable to encounter resistance problems and to limit crop losses should fungicide resistance appear. So if you look at this label on the bottom left, this is for broadworm, which is a group 711 fungicide. So FRAC assigns numbers to fungicide active ingredients based on their mode of action, or that's the way that fungicide is targeting botrytis. So for example, on this label, we have group 711, and 7 corresponds to fluopyram, which is your group 7 active ingredient, and 11 corresponds to trifloxystrobin. It means that both of these active ingredients are targeting different parts of botrytis. And this is important because later on, you'll realize that we have a lot of 711 mixtures on the market, at least in Michigan. And Sarah's probably the best person, or perhaps your local educators, to ask about products that are registered in Ontario or your region. Here is an example of some of the common Michigan products that contain ingredients with similar FRAC codes or 711s. So on the left side here, the left column is the product name, the middle we have active ingredients, and the right is FRAC codes. If you look at the middle, we do have a wide range of active ingredients. However, they all share 711 or a 711 mixture. If you continue to apply a 711 mixture over and over again, Botrytis is going to sort of get used to that fungicide and it's going to be able to overcome it, and the fungicide is no longer going to be effective. So to give a little bit of an outline of how fungicide resistance becomes widespread in a greenhouse, if you look at the far left green square over here, this represents a greenhouse with Botrytis. And all of the black ovals here are going to represent strains that are sensitive to a fungicide, for example, maybe Decree. So most of the Botrytis you have in your greenhouse will be sensitive to Decree. You may have some that's resistant, but that's naturally occurring. But if you apply Decree here, you're going to eliminate most of the sensitive isolates in your greenhouse. There may be a little bit still left over from part of a plant that didn't get exposed to the fungicide. But if you can continue to spray this over and over and Botrytis reproduces, eventually your greenhouse is going to contain all the Botrytis that's resistant to Decree. And next time you go and try to apply Decree to manage Botrytis, you're not going to get any control. So moving on, now we're looking at some of the trials that Blair ran. This is Botrytis on Poinsettia, early prestige red, looking at similar FRAC codes. So your 711, 711 mixes. On the left side here, the y-axis is the flower disease severity. So a 0 represents a healthy plant and a 10 would be extreme Botrytis blight. So on the left, if you're looking at your untreated, here we have high disease severity, unsurprising because we didn't apply anything to manage Botrytis. And if you move to the left of that, we have Picotina, Picotina flora, Palladium, Orchestra intrinsic, and Punctellus. And what the takeaway really is here is that all of these bars share a letter in common. They all have an A, compared to the untreated with has B, which tells you that Picotina, Picotina flora, etc., all of these are effectively controlling Botrytis, and there's no significant difference between them. So if you were to apply Picotina at 10 fluid ounces on a 7-day interval, you're going to see about the same level of control as you would applying Punctellus 24 fluid ounces on a 7-day interval. So all of these are effective at controlling Botrytis. So moving on, here we're looking at different products that provide similar control. Again, here we have Emblem, Orchestra intrinsic, Aston, Mural, and Affirm. So the left side here, we're just looking at leaves and disease severity. Same takeaway as the last graph. All of these have a lower disease severity than the untreated, which means they're providing control against Botrytis. Here's some pictures for you to visualize what we're talking about. On the left here is no fungicide. You can see that plant is pretty much a goner. I do want to let you know that when we run all of these trials, we're taking fully grown poinsettia plants and we place them into baskets within a plastic bag and a lot of water. So we're basically putting these into what we call a torture chamber. It's the worst conditions for Botrytis possible. So we get the highest disease pressure possible, and these products are still doing well, which tells you they're pretty effective. So on the right here, we have Emblem, 4 fluid ounces at a 14-day interval, did a good job at managing Botrytis. Moving on here, we have Orchestra intrinsic and Aston. Again, like I said, no significant difference between these. They're all providing good control. And this is continued with Mural and Affirm. So moving on now, here we're looking at Botrytis on Geranium. This is Pinto Premium White, and we're comparing intervals 7-day versus 14-day. And again, on the left side here, y-axis is percentage of sporulating leaves. So the higher the bar, the worse the disease. Unsurprisingly, your untreated inoculated is pretty high. The 2 sections that are missing here, I just took out because they're not registered. If we make this a little bit more simple here, first we're looking at Palladium. On the left side is a 7-day interval and the right side is a 14-day interval. And what you'll notice here is that they still share the letter B in common. So there's no significant difference between applying at a 7-day versus a 14-day interval. They're both providing more significant control than your untreated. So next here, we're looking at pageant. We have 14-day interval on the left and 7-day interval on the right. Again, they both share the letters A, B, both providing good control. You're not going to get better control by applying at a 7-day interval than you would 14-day. And again, pictures to help you visualize this. Left is 7-day pageant, right is 14-day. Pretty good control. So now we're looking at different rates. Botrytis again on Geranium, Pinto, Premium, White. More is not always better. So again, y-axis on the left side is the sporulating leaves. The higher the bar, the worse the disease. On the bottom here, we're looking at broad form 8 fluid ounces, broad form 4 fluid ounces. Decree is a control, and Aston at 17 and 10 fluid ounces. And the takeaway here, right, is that they all share the letter A and they're all lower than your untreated. So you're getting good disease control with broad form and Aston, no matter what rate you're applying. There's no significant difference between 4 fluid ounces and 8 fluid ounces. However, like I said before, if you try to go even lower and apply 2 fluid ounces per se, it's possible you won't get any disease control. So again, pictures for you. Untreated on the left here, we have botrytis lesion throughout, broad form on the right looking pretty good. And same thing here with Decree and Aston. So while I will give fungicide recommendations in a little bit, I first wanted to talk about the biopesticides that we have available in Michigan. So the left side here is products. We have a wide range, active ingredients in the middle, and FRAC codes on the right. So remember, if you're applying a lot of 7-Elevens, if you want something else to throw in there to help manage resistance, maybe look to biopesticides, especially when you have lower disease pressure. Some of these are not super effective, as you can see here. So left axis again is percentage of sporulating leaves, and we have our untreated on the left side. If you go to the middle and you look at the lupinus extract and Botector, both of these bars share the letter D in common with the untreated, which means that if you apply lupinus or you apply Botector at these rates and intervals, it's going to be no different from as if you did nothing. So really a waste of time here. If you're applying Prophytex or Proud3, these columns are even bigger. So we had worse disease than the untreated. So you're better off doing nothing than applying those. And then if you look at the left here, Stargus is the only biopesticide tested in this trial that effectively controlled botrytis. A little bit of disease, but still did better than our untreated. And pictures to drive this home again. Left here is Stargus, doing okay, not perfect, but if you have low disease pressure, this would probably be a good tool because like I said, we're throwing these in plastic bags with water to get as much disease as we can. Lupinus extract on the right side. For a good portion of that plant is covered in botrytis. Then we have Botector and Prophytex, which are not looking too great. So moving on now, I want to talk about some of the research that I've been doing in our lab at Michigan State. The first one I'm going to talk about is the screening for fungicide sensitivity in Michigan ornamental greenhouses. So as Jeremy said, I started collecting back in 2018. I went around with extension educators and we went to the major production regions in Michigan, visited about 20 floriculture greenhouses, some of them up to 4 times. And I sampled from 3 different crops, looking at poinsettia, geranium, and petunia. And I really tried to get a wide range of greenhouses, crops, years, etc., to really get a good sample size. And in the end here, I had about 339 plates of botrytis, which is what you see on the right here, that I used to screen for resistance to a panel of fungicides. So here we're looking at the fungicide sensitivity screening in the lab. On the left side of this over here is the active ingredients I looked at. So we have thiophanate methyl, iprodione, boscalid, fluopyram, cyprodenil, pyraclostrobin, fludioxonil, and penhexamide. And the goal here was to get a wide range of FRAC codes, as you can see, because, for example, if you have resistance to pyraclostrobin, which is a FRAC 11, it's likely that you're going to have cross resistance to other fungicides within that FRAC 11. So azoxystrobin, trifloxystrobin, etc. So I tested one of that FRAC code. In contrast, you can see that I have 2 from FRAC 11. That group's a little trickier, so I included both boscalid and fluopyram. And this picture on the right is what it looks like in the lab. So this is what we call a 12-well plate. And in each of these wells contains a basal media, so just some sugar that Botrytis needs to grow, and it may contain a fungicide as well. And I can compare the wells with and without fungicide to see whether or not these isolates are resistant. And if I look at these under the microscope, here's an example of what I see. So top left picture, here we're looking at Botrytis, which is those little gray dots you see in there are the individual spores, and they're germinating and growing well inside a well without fungicide. However, if you add fungicide to that and go to the right, the top right picture here shows those spores are not germinating, not growing. The fungicide is effectively stopping growth, and we would call that a sensitive isolate. In contrast, these pictures on the bottom show you that this isolate doesn't care whether or not there's fungicide, it grows just as fine. And here we have an example of resistance. So after screening all 339 to that panel, here is the results that I saw. So in this picture here, the y-axis is fungicide resistance frequencies. And this is the percentage of the 339 isolates I looked at that were resistant to the particular fungicide on the bottom x-axis here. So if we start on the left side, we're looking at thiophanate methyl. And I found that about over 90% of the isolates I looked at were resistant to thiophanate methyl. So for a while now, we have not recommended this fungicide for botrytis control, and you can see why. We move to the left of that, and we have pyraclostrobin, iprodione, and boscalid, where over 50% of the isolates I looked at were resistant to these fungicides. Then we have some intermediate levels of resistance to cyprodenil, fenhexamide, and less resistance to fludioxonil and fluopyram. Fluopyram is not too surprising because that is a newer fungicide, as there's a lot more FRAC 7 products coming out recently. I might expect this resistance to become more common with time. So next I want to tell you about chemical class resistance, or CCR. Again, going back to FRAC, FRAC provides recommendations that you need at least 5 different modes of action, or 5 different FRAC codes, per crop disease combination in order to ensure effective resistance management. So this means you need 5 different FRAC codes or 5 different products if you want to prevent resistance from developing in your greenhouse. CCR comes in because it stands for chemical class resistance. This is the number of chemical classes or the number of FRAC codes that an isolate is resistant to. So for example, CCR2 means that the isolate is resistant to 2 FRAC codes. Maybe it's resistant to FRAC 7 and FRAC 11, for example. I try to show you results from this. Just to sum up what I'm getting from this picture, on the left side, we're looking at 3 different crops. We have Poinsettia, Petunia, and Geranium, crops I sampled from. And the colors in each of these bars represent the proportion of isolates that are resistant to a specific number of chemical classes. So I know it gets a little tricky, but hang in with me. Top right here, for example, if you're looking at this dark red section here, this is the proportion of isolates I found that were resistant to 0, so sensitive to all 8 fungicides I tested, all 7 chemical classes. So a really small proportion. So it shows you that it looks like less than 5% of the isolates I screened were sensitive to all fungicides. And in contrast, if you go to the far left here on Petunia, for example, that dark gray area represents the proportion of isolates from Petunia that were resistant to all 7 chemical classes or all fungicides that I screened. So you can see here that no matter the crop, Poinsettia, Petunia, Geranium, you have a pretty even ratio with a lot of resistance going on. If you look at the dark gray bar here, the third one that represents 5 going down the middle, it looks like almost 50% of the isolates I have are resistant to at least 5 chemical classes, which means there's a lot of resistance in these ornamental greenhouses. So knowing all of this, we'll move on to fungicide recommendations. These recommendations are based on years of product efficacy trials in Michigan greenhouses, many of which conducted by Blair Harlan in our lab. And the fungicide resistance screening I've been doing within major production regions of Michigan in the last 4 years. So in our lab, we usually give fungicide recommendations using the analogy of the A team, B team. So A team is going to be your first line of defense. If you have a lot of disease pressure and you need really good control, these might be the products you would go to. We have a wide range of FRAC codes and brackets on the end here. So for example, Affirm with the active ingredient of polyoxidized zinc salt belongs to FRAC 19. And you can see we have a good list here. Again, a few 7-Elevens in here, Broadform, Mural, and Orchestra. So if you are doing a rotation, it would be a bad idea to rotate Broadform with Mural with Orchestra, because eventually Botrytis is going to become resistant to both of those. And you're going to go to apply Broadform and get zero control. Moving on now, we're looking at the Botrytis B team, and this is your second line of defense. Pretty short list, just includes Iprodion and Pageant, which is a mix of pyroclastrobium and boscalid. We have 2 different FRAC codes here, and really when you're applying these, you should have low disease pressure in your greenhouse. These products aren't the best, but they will do well if you don't have a lot of disease. And if you're looking for these list of recommendations at the very end, I'll provide our website where you can find all of our fungicide recommendations. So moving on, the next research I've been doing in our lab is to evaluate the ability of foliar calcium chloride applications to protect against Botrytis blight on ornamental crops. So to give a little bit of the background here, we're talking about calcium as an alternative to fungicides. So weaker senescent plant tissue can be a result of calcium deficiencies. If you look at this picture on the right, we're looking at bract edge burn on Poinsettia. In some cases, they can attribute this to to calcium deficiency. And this portion of the plant is senescing and becomes a target for Botrytis. We have high relative humidity and overcast conditions. These are going to slow calcium translocation, which means that calcium deficiencies, especially in upper plant parts, is going to be more common. And this environment is the same environment that's conducive for Botrytis, the high relative humidity, overcast, lots of free moisture in the greenhouse for Botrytis. So knowing all of this, growers have been applying foliar calcium applications to reduce Botrytis incidence in their greenhouse. So I went through now and looked at a few different rates of calcium. We looked at calcium chloride with and without fungicides. And here's the results I saw. So left column here is disease severity. The lower the bar, again, is a healthy plant. 0 is healthy. And 5 represents severe defoliation, lots of Botrytis. If we start on the left over here, the second to last bar, we're looking at untreated. These plants had a really high disease severity rating. And if you look at the letters here, C, and the letter in the column to the right, which is just calcium, also C. So it looks like if you apply calcium at a rate of, this is 16.7 ounces, which corresponds to just about 350 parts per million, which is a rate that many growers have been applying. You're not getting any disease control here. It's as if you're not doing anything. And to the left here, we looked at Decree both with and without calcium. And again, this didn't do anything. And you'll notice that while Decree was on our A list of products, the particular isolate I used in this experiment was resistant to Decree. So these results are not surprising. To the left of this, we're looking at palladium with and without calcium. They share the letter B. So there's no significant difference if you're applying palladium by itself or palladium with calcium. Same thing with broadform. Broadform with and without calcium shares the letter A. So it looks like while these fungicides are providing control, the calcium does not have an additive effect. And even by itself, it's no different from the untreated. Here are some pictures for you to visualize that. The middle, we have untreated, really unhappy plant, Decree as well, again, because this isolate was resistant to fenhexamide, which is the active ingredient in Decree. The bottom here, we're looking at calcium, which did not do much to protect from Botrytis. On the right side here, we have palladium in broad form, good standards. So I said, we applied this at 350 parts per million. What if we up the rate? So we looked at this at 400, 800, and 1200 parts per million of calcium. And as you can see here, disease severity the same. The lower the bar, the healthier. We have untreated on the far left, which is about 3.5. And our 1200, 800, 400 are all at 5, which means they did significantly worse than our untreated. So even if you're increasing your calcium rates, you're still not getting protection from botrytis. And here are pictures to show you that. None of these are looking very great. So to sum up today, we talked about how you can lower disease pressure by reducing relative humidity in periods of free moisture. Remember, below 85%, watering early, venting, heating, fans, all of that can help with this. It's important to follow the rates and spray intervals found on fungicide labels, although it appears that rates and spray intervals do not have a significant difference. Fungicide resistance is common along all the crops that I tested. All the ornamental crops, we're looking at Poinsettia, Geranium, and Petunia, and it appears that there's no significant difference in resistance among them within Michigan. We talked about how it's important to rotate those FRAC codes so you can elongate product efficacy and basically help to prevent resistance from occurring faster. And at the very end, we mentioned how foliar applications of calcium fluoride are not providing significant protection against Botrytis cinerea on Poinsettia. I'm running trials right now in our greenhouse, looking at this on Petunia, New Guinea impatiens, and Geranium, but tentatively we see the same results. So with that, I would like to thank the Floriculture Initiative of the USDA Ag Research Service, American Floral Endowment funding sources, as well as the Metro Detroit Flower Growers Association, Western Michigan Greenhouse Association, and our MSU extension agents, such as Jeremy. Thank you.
Great, thank you so much, Nikki. That was really informative. We've got a couple questions rolling in the Q&A function. I'm going to start with a few questions of my own and give time everyone else a bit more time to put them in the Q&A section. I was wondering whether botrytis, I know mainly it's a foliar disease, but it can affect stems, and I understand it can get into the roots. Can it be spread through recirculating water at all, or is it mostly through ventilation and blowing and lack of sanitation like you were explaining?
Definitely ventilation, blowing, lack of sanitation.
Okay.
I don't know if there's been any research. If so, very little of it traveling through water sources. That's unlikely.
Okay.
And
well,
usually,
one less thing for growers to have to worry about.
That's true. And when you hear about it targeting stem and root, that's often a secondary cause. So
Okay.
maybe you have Pythium causing root rot, and then once the Pythium dries up, maybe you get botrytis coming in as a secondary problem.
Okay, that makes a lot of sense. You were talking about those leaves that drop and then get the spores on them as a source of inoculum. What is the best way to remove them from the greenhouse? I've heard because they're so delicate and blowy that you should maybe put them in a wet paper bag and fold them up so that you're not walking around just like with an open dry bag and they can go everywhere. Do you have any recommendations on that?
Here's the problem. A good answer there is to try to apply a protectant fungicide before you're going to go and move all your plants around. This goes the same with propagating or before shipping. Good idea to apply protectants before that because you know botrytis is going to be moving around no matter what you're doing, really.
Awesome, that's a great idea. We have a few questions around the biofungicides, which I was really interested in. It was really interesting to me that only Stargus was the one that showed sort of any effect. Do you know why that is? Is it the specific bacillus species or specific strain? Why don't the Bacillus subtilis work? Are you going to test different microbials than the ones you tried here? Just to give us the whole answer on that.
Yeah, a little bit tricky. Obviously, this is going to depend a lot on environment. And like I said before, while your biopesticides may not be as effective as conventional, we are throwing these plants in those torture chambers where you have really, really high disease pressure. And while these may do well in a greenhouse where you don't have as much disease pressure, in these conditions, oftentimes they're not going to be ideal. So
maybe they might have a role in sort of protection under low, pest
my guess
pressure,
is they would do better under low disease pressure.
Yeah, I like your analogy of a torture chamber, which is basically what a lot of grad work is. It's like, let's do worst case scenario, see what happens.
So this is worst case scenario, that biopesticides aren't going to do great when you have the worst conditions. It doesn't mean they won't do anything when you have light disease pressure.
Another follow-up question for that was whether you saw any resistance among any of the biological treatments, or just haven't had time to follow that up yet.
We haven't screened for that before. I don't think it would be as much of a problem because they work differently than our conventionals.
Yeah, that's my understanding as well. Another question, just to guess about how you did your tests exactly. Someone's wondering what the source of inoculum was that you used, i.e. a single isolate collected from the same host species or a mixed population?
In most cases, yes, this was a single isolate. Generally, that's because you can get mixed results if you have a mixed population in these experiments. But again, in all of these experiments, at least for a lot of them, we use different isolates between experiments, even if it wasn't in the same one. So if your question is about maybe the calcium or something like that, if you use a different isolate, you may see slightly different results. It's possible.
Great. More questions rolling in. This is really popular. This is great. Thanks everyone for putting in your questions. This is wonderful when we get to have this level of engagement with the speaker. You mentioned the calcium really didn't do much, which was obviously a little bit disappointing. Any work done using silicone to build plant resistance to infection?
I have not, but I know there's been a little bit of work. I think that came out of Clemson University, and I don't know if they saw, can't remember if they saw some phytotoxicity problems or something that was secondary with that. But I haven't done any work on that myself, so I can't say much.
Okay, I mean, you may have said it in your talk, but I think I might have just missed it. What was the rationale for picking calcium in the first place? I recall you said that the symptoms can mimic calcium deficiency, so maybe if you add them together. Was there any other things in the literature that showed that sometimes adding calcium can prevent disease?
So there's two different ways that they hypothesize calcium works. The first of this would be strengthening the plant cell wall. So like I said, when you have a calcium deficiency, this leads to senescent tissue, oftentimes just because your plant cell walls are breaking down easier. So the theory behind this is if you're applying calcium, you're strengthening the cell wall. And because botrytis is generally considered a weak pathogen and targets weak tissue, that those stronger plant cells will be less susceptible to botrytis. The theory behind it, but as you can see, obviously not that well.
Great. We've got a few more questions. I'm so impressed with everyone. Thanks. These are really insightful questions too. Someone is asking, they know that you said that diseased plants serve as an inoculum source, but they're wondering if the aerospores are ubiquitous. What's your opinion on the degree to which the inoculum load versus humidity control plays a role? So similar to removing source plants, which you recommended, how do you think something like scrubbing aerospores from the greenhouse air to reduce disease incidence would work?
Where is this? Oh, I'm looking for this question.
That one's in, yeah, that was sort of bad reading on my part. That's in the Q&A. It's the second question down.
I see. It's a long one. All right.
Yeah.
So what is your opinion on the degree to which inoculum load plays versus humidity control? So honestly, I would say humidity is a much larger player here because again, even if you have a lot of botrytis in your greenhouse, but you have low humidity, you're not going to have disease. If you have humidity and you have one leaf per se, that can blow up very quickly because symptoms can result in 24 hours and you can have this cycle going over and over very quickly. So it blows up very quickly under high humidity and free moisture.
Yeah, and I remember you saying that cutoff was around 85% humidity is the where you get into the scary bad zone. So
yes.
Excellent.
And I think they also asked, and similar to removing source plants, would scrubbing aerospores from the greenhouse air reduce the disease incidence?
Yeah.
I'm not sure what scrubbing aerospores from the greenhouse air means.
I think that's a project we're looking at, that's basically a dehumidifier that's also sucking spores in and killing them and then pushing clean air out. So
Mhm.
I guess they're just wondering how many like free spores are floating around the greenhouse environment, and would that help?
Well, I can tell you that a lot more spores are going to be around in the air when you're moving plants around, but we haven't done any work on using filters or anything of that sort.
Yeah, and good point about when you're moving plants around, that's another time when things can stir up and cause more problems. Let's see. Someone was wondering if you tested the plant tissue to see if the calcium content was increased with the applications you made. So was the plant actually taking it up?
So good question. I didn't mention it, but we did apply calcium on a foliar basis for several weeks prior to the actual inoculation point. So in theory, we would expect it to, but actually measuring the calcium concentration would be a much more in-depth experiment. And this has been done in other research. And they have shown that, yes, calcium has increased, but this does depend on tissue type as well. So a lot of calcium work has been done, I believe, on apples and pears and fruits, not as much on the ornamental side. And it's also the side where you have more variation in tissue types. So it's tricky to do.
Yeah. Last question on this end, and then I'll see if any of our hosts have any questions from their floriculture experience. Jamie's asking if there's a way to spray preventatively for botrytis. Not necessarily that we promote that approach, but would spraying fungicides or biofungicides be more effective if sprayed preventatively rather than curatively?
Good question. I would say, while this does depend on the product, I would say generally yes. So products are per se labeled based on their activity. So some of these can be just protectants where they just sit on the surface of the leaf. And some of these are more of systemic fungicides that are taken at fungicidal levels and spread throughout the plant. So while this does depend on the product, generally preventative is much more effective than curative, because once you have botrytis in the plant, none of those protectants are going to be able to get to it. So preventative is better, but not necessarily if you want to be applying less fungicide.
I'm just wondering how you would balance that with the whole A team, B team idea that MSU uses that I really like, but we obviously have a lot fewer products on both our A team and our B team. So for a preventative application, would you pick a B team and try to keep your A team in your back pocket for when there is more of a problem? Or would you start with an A team and hope you have a second A team of a different FRAC number to move to if you needed to?
Depending if you're applying preventatively because you're moving your plants around, for example, then yes, I would say go with the A team. A lot of those products are going to have more residual control. For example, I think botrytis has shown to provide protection for several weeks following initial spray time. So in contrast, if you're going to apply a biofungicide and then you have botrytis introduced to your greenhouse 2 weeks later, it's possible that that's not doing much at that point. While in contrast, you may want to apply a biofungicide rather than, say, broad form or some other 7-11 mixture, or if you don't have 7-11 mixtures, a 7 or 11, it's possible that that product may have more residual control and still work after a few weeks. So you win some, you lose some. Never perfect.
Yes, it's always a balancing act for sure. Siobhan and Jeremy, do you have any questions before we wrap up?
I found one more that came in actually.
Oh, great.
So this one says, is water quality and water health also helpful in preventative measures, or is it really more related to just the air quality?
I mean, indirectly, both of them, yes. The healthier your plant, the less likely you're going to get botrytis. Directly, not so much.
I think I'll throw the last question out then, Nikki. So thanks so much for telling us all about your research today. And I'm assuming that you're probably wrapping up your projects, but can you tell us a little bit about what future work the lab has planned to look at botrytis issues that might help our growers?
Yeah, so I'm hoping in the future we'll continue with our fungicide resistance screening. It would be nice if we could look more into the FRAC 7 products that are coming out. I'm not sure if they're coming out in Ontario as well. Do either of you?
I'm not sure yet, but we can look into that for sure.
More FRAC 7 products coming out, continue with fungicide resistance screening. And of course, there's a lot more interest in biopesticides. So we do contract trials. I'm running about 8 of them right now in our greenhouse, testing about 30 different products. So we're always testing new products. I have a lot more information that you can't fit into a 40-minute talk. But yeah, we're always testing more.
Excellent. Well, thank you so much for being willing to come and talk to a Canadian audience and share your very holistic view of botrytis control, which I think is really important. And we look forward to hearing more of your work from the future. It sounds like you're doing a lot of exciting things with biologicals and new products that Canada loves to get ideas from the US. So yeah, we'll be hearing more from you in the future, I'm sure.
Great. Thank you very much.
Okay, and we're just going to quickly share our screen and do some shameless blog promotion. So let me see if I can get to the right slide. Maybe not. Oh, too far. There we go. Siobhan, if you want to explain what on floriculture is and why people should care.
Yeah, so we mentioned this at the beginning, but once again, Sarah and I run a blog where we post lots of information about things that are going on in the floriculture sector in Ontario, issues that we might have heard about or that growers are experiencing. We advertise our own webinars and other webinars you might be interested in, and we share lots of resources. It's a great place to go to. You can also find all the recordings of our previous GrowON webinars on our blog. So if you're not subscribed, please go ahead and take a look. It's got an easy address to Google. It's on floriculture.com.
Great. And when we end this webinar, there's a survey that's going to pop up. It would be great if everyone could fill it out. And like I said, if you missed some of the webinar, if you came in late, the recording will be up afterwards. So thanks everyone for joining and have a great rest of your Thursday.